So we're here with Dr. Li Zhu. Well, he is the professor of pathology at Harvard Medical School, as well as the an associate scientific director at Massachusetts General Hospital. So Dr. Zhu, can you tell us a little bit more about what your research focuses on?
Yes, so I'm a cancer biologist, but actually I'm a basic scientist. So because in cancer biology, we have basic scientists and also clinicians working on all kinds of important problems. And I'm actually one of the basic scientists here in the meeting. So my main research interest is trying to understand how cells detect different type of DNA damage, different lesions in DNA in the genome, and how cells respond to them and eventually repair them. This is actually quite relevant to ovarian cancer because, as you know, BRCA1, BRCA2 genes are highly mutated in familial breast cancer patients. And it turned out that BRCA1 and BRCA2 both are important DNA repair genes. So we study them very extensively in our project, try to understand how they normally function and what happened to them in cancer cells, in ovarian cancer cells, when they're mutated, when they're defective, what's wrong with the DNA repair process, and how would that provide an opportunity for therapy. So that's actually one of the main research focus on my lab.
And a topic that seems to be pretty hot right now is molecular profiling.
Mm-hmm.
So can you tell us a little bit about your opinion on how that might guide treatment decisions and what our survivor community would need to know about that?
Right. So the reason molecular profiling is important now, especially for BRCA1, BRCA2 mutation carrier patients, is that now there's a drug called PARP inhibitor. So PARP is a protein involved in DNA repair.
Right.
And what scientists have found is that when you inhibit that particular protein in patient cancer cells without functional BRCA1, BRCA2, those cells become very sensitive. They're very sensitive to the drug. They're being killed selectively by the drug. So there are several PARP inhibitors have been approved by FDA for the treatment of ovarian cancer. So this is why. So if a patient knows she has mutations in BRCA1, BRCA2, there's a good chance that the tumor will respond to PARP inhibitor. So that's why molecular profiling could be helpful now. And also, molecular profiling may be important during the treatment. Molecular profiling may be important to determine if resistance will occur and what kind of mechanism will be underlying the drug resistance. And this perhaps is important for making decision how to overcome drug resistance to PARP inhibitor later on. So this is something that's still being developed, but it's a kind of a very promising cutting edge of the ovarian cancer research now.
Yeah, that sounds pretty promising at
Yes.
this point. You said it's in the early stages.
Yes.
How early are those stages?
Yes. So right now, it's still very much basic research. People are still trying to understand how cancer cells, ovarian cancer cells with BRCA1, BRCA2 mutations, how they develop resistance to PARP inhibitor. So a lot of basic research labs, including my lab, are working on this problem, try to understand different mechanisms that could lead to resistance, and eventually try to come up with strategies to overcome resistance. So a lot of research are actively going right now. So some of the ideas are in clinical trials now. For example, combination PARP inhibitor with some other drugs, including CHEK1 inhibitor, WEE1 inhibitor. So these are the ideas people kind of came up from basic research, try to overcome PARP inhibitor resistance in ovarian cancer with BRCA1, BRCA2 mutations. And actually, one of the projects in my lab is exactly that, try to understand how cells develop resistance and how to overcome that. And so what we found is that we work on a kinase, a protein kinase called ATR. So ATR turns out to be a very important regulator of DNA repair processes.
Mm-hmm.
So in those cells with BRCA1, BRCA2 mutation, so those cells are very sensitive to PARP inhibitor. But when they develop resistance to PARP inhibitor, cancer cells are very smart. They can actually rewire their DNA repair pathway to bypass BRCA1, BRCA2. So
They're like hackers.
Exactly,
Very
exactly.
annoying.
Very annoying, very annoying. So now we're trying to understand their strategy, basically.
Right.
Yeah, their strategy is to so bypass BRCA1, BRCA2. So now cells don't need them, and they don't care if PARP is inhibited or not. So what happens is that when they rewire their DNA repair pathway, they become more dependent on this kinase called ATR, because that ATR kinase is required for that alternative pathway in the absence of BRCA1 and BRCA2. So now the idea we're trying to propose is that when BRCA1, BRCA2 cancer cells become resistant to PARP inhibitor, so we will treat them with the combination of PARP inhibitor plus ATR inhibitor. So this way, we're blocking that alternative pathway in those cancer cells. So now they will become sensitive to PARP inhibitor again. So this is the idea we're coming up with. But still, it's still a little too early. So we're still doing basic research on this idea, try to understand how exactly ATR functions in those resistant cells, and also trying to collaborate with clinicians to initiate clinical trials to test this idea eventually in patients. Yes.
Okay, so you touched on my next question here, which is basically a majority of patients are seeing resistance in PARP inhibitors. Yes. And if you could tell us a little bit more about the approaches in practice and what's being developed to help overcome those things.
Yes, yes. Yeah. So cancer cells are very annoying because they have many ways to become resistant. That's really the problem.
Okay.
So for example, right, so when they become, when BRCA1, BRCA2 mutant cancer cells become resistant to PARP inhibitor, so people have learned in the past few years that they actually use different strategies. Yeah, there are several ways they can become resistant. For example, they could have additional mutations in BRCA1, BRCA2 to make the mutant kind of back to more like the normal protein. Yeah, they have secondary mutations kind of to restore the protein in some way. So that's really annoying. Yeah.
Right.
So because now you have, the idea is to target cancer cells with defective protein, but now the protein is not so defective anymore. So
they've camouflaged itself into looking normal.
Yeah. So that's one of the mechanisms cancer cells could become resistant. And the other mechanism is, as I mentioned, they can try to rewire the pathway somehow just to get around that BRCA1, BRCA2 mutation. Now they kind of restore their ability to repair certain DNA damage, so they're not sensitive to PARP inhibitor anymore. So there are specific proteins people have found that when they're mutated, they actually bypass BRCA1, BRCA2. So for example, a protein called 53BP1, another protein is called PTIP. So when these proteins are mutated in those cancer cells, they confer resistance. So these are the problems. Yeah, so one idea now, right, to overcome resistance.
Yeah, I'm curious to know. It's like whack-a-mole. What are you doing to
exactly.
get around this?
So this is kind of the way we're thinking about it, right? So yes, there may be many ways to rewire the DNA repair pathway, but eventually those cancer cells still need DNA repair. Yeah, even the pathway could be very different.
Right.
So the idea is to find something downstream, meaning that they function normally kind of after BRCA1, BRCA2 in the repair process. So if you have kind of a process, right, you need BRCA1, BRCA2 at some point during this process. When cancer cells kind of bypass them, they go around, but they need some of the factors kind of downstream at the end of this process to carry out repair.
Right.
So we try to find those proteins, what they are, and hopefully we can target something there. And it turns out ATR is probably one of those factors. And now there's a drug to target ATR. It's already in clinical trial, phase 1, phase 2. The idea is to combine that ATR inhibitor with PARP inhibitor. So we hit it at the bottom of the process. So even BRCA1, BRCA2 are bypassed in those resistant cells, but we can still find that.
Get it at the root of the
exactly,
problem.
getting to the root. So that may be one way to do it.
Okay.
That may be one way to do it, yeah. So the other thinking is that, so sometimes, BRCA1, BRCA2, they have this kind of secondary mutation somehow restore the function. Yeah. So the idea is to somehow, if you can reduce that function again, yeah. So cancer cells are trying to get the function back, but if you can have drugs to push those functions down again, yeah, you can resensitize them to PARP inhibitor. That's another way of thinking. And again, so people are looking at targets that regulate BRCA1, BRCA2 function. If you inhibit those regulators, so even when BRCA1, BRCA2 functions are partially restored, you can still kind of press them down, and cells may become sensitive to PARP inhibitor again.
Yeah.
So those are the line of thinkings. Yeah.
It's
So
really
people are
interesting.
thinking,
It's almost like you guys are creating a military strategy to go in
exactly.
and
Yes.
attack. Yeah.
Very similar to that.
Yeah.
Very similar to that.
So hopefully
yes.
we'll win all these battles so we can win the war, right?
Yes. Yes. That's the hope. Yes.
So can you tell us about the new knowledge that's emerged through research in terms of DNA with damage and repair metabolic changes in ovarian cancer cells and tumor microenvironment?
Yes, yes. So my expertise is really in the DNA repair aspect, but a lot of things are connected, right? So for example, DNA repair is heavily influenced by metabolic changes in cancer cells, and it is also heavily influenced by microenvironment. So that's actually kind of a new area of research to see how these different things interact with each other and affect each other. Yeah, because in a cell or in the cancer cell, everything is connected. So right now, a lot of studies are focused on specific areas of kind of biology in cancer cells, but things are connected. So this is a new trend. Yeah, scientists now are looking at different processes in cancer cells as a group instead of as individual events. That's what we hope to do, and many people are hoping to do that now. But in terms of DNA repair, right, so even as an independent event in cancer cells, it is really important because now we have drugs to target it. Yeah, other people are also developing drugs to target the metabolic processes or alterations in cancer cell or the alterations in microenvironment in ovarian cancer tumors. All those are important. So the hope is eventually we can come up with magic combination of drugs targeting different aspects of the cancer cell biology and to achieve more specific killing of cancer cells.
Yeah. So you guys are basically on the hunt for like this algorithm to tear down this hacker,
exactly,
right?
Exactly.
Okay. I've turned this into a whole science fiction movie, but
it's exactly like that.
Yeah.
Exactly like that. Yes.
So how is the knowledge being applied with this DNA repair being applied to more effectively control ovarian cancer?
Yes. Yes. So the first successful example, this is PARP inhibitor.
Yeah.
Yes, because that's a classic example. Yeah, you have a defined kind of mutation. So if you find mutations in BRCA1, BRCA2, then those tumors will have a good chance to be responsive to PARP inhibitor. So you have very clear genetic alteration, and a drug can respond to that. So that's one great example. So people came up with a term called synthetic lethality, meaning that if a cancer cell has specific defect, right? So for example, BRCA1, BRCA2 gene mutations, and then you introduce a second defect using a drug. Yes, in this case, it's PARP inhibitor, so you're inhibiting a specific DNA repair process. Synthetic lethality means that two defects are incompatible. When you combine them, you're killing the cells. And this only happens in cancer cells because cancer cells have those mutations. So they're particularly sensitive to the drug.
So you put two negatives together and you get a positive
Exactly. So this idea is still very attractive. So now what people are trying to do is to find other drugs. Yeah, when you combine the drugs with specific genetic defects in cancer, and in this case, particular in breast cancer, you will have this specific killing of cancer cells. This is the idea going forward. So people are trying very hard to do that. And this idea is not only good for targeting DNA repair defects, such as BRCA1, BRCA2 mutations, as you mentioned, right? So cancer cells also have alterations in metabolism. They have changes in different metabolites in cells. And that may also provide an opportunity for intervention, for drug targeting. But it will be different mutation and different drug. But the idea is the same. that cancer cells are already defective or something, and now if you introduce a drug that inhibits something else, that combination will be lethal specifically in cancer cells. Yeah.
And how does the understanding of mechanisms of resistance help in developing the approaches to address the challenges when it comes to the resistance of PARP inhibitors?
Yeah. So very important question. Yeah. So this kind of I think it will be a process to really understand the resistant mechanisms and eventually come up with drugs to overcome them. So I think there are several steps that people have to accomplish in this process. The first step is really to understand how resistance arises.
Right. Know your enemy.
Yeah, exactly.
Okay.
Yeah. So what we're learning now is there are multiple ways cancer cells could become resistant. And we have to understand each mechanism of resistance really to design strategies to overcome them.
Right.
Yeah. So this is important, step one. And step two is to find the right drug target to overcome it, and to have to develop useful drugs to do that. That's also a very important step. So for example, people have found factors that may be important in BRCA1, BRCA2 mutant cells when they become resistant, but we don't necessarily have all the drugs to target those factors. It will take time and a lot of effort to develop new drugs to target those potentially useful targets. So that's another kind of important step in the process. And eventually, I think that very importantly, so we have to have good clinical trials to test the combinations of PARP inhibitor and those new drugs in patients that develop resistance. So this is again, it's a group effort, basic scientists, clinicians, and patients too. Yeah.
Yeah, it's always more heads better than one.
Yes, absolutely.
So I know that it's still you said in clinical trials, and then, but how far away is this really? You know, as a survivor community, you're wanting to know, yeah, you guys are working on this, but how long before this is something that, you know, is very viable to me as you know, someone that's suffering from ovarian cancer?
Right. So PARP inhibitor, right? So obviously, it's a FDA approved drug, is already being used, but it will take a little bit time to actually develop this combination PARP inhibitor plus something else. Right, so one of the limiting factor is actually this second drug. A lot of this second drug are still being developed. So for example, ATR inhibitor, so something we're working on, is in clinical trial in phase 1 and phase 2, but it's not FDA approved yet. So it will still need some time and extra clinical trials to test this drug to make sure it's safe for patients to take, and this combination is actually effective in patients. So yeah, it's a great idea. I think it's a great idea based on the basic research, but still it will take a little bit more time for the drug to be approved by FDA and eventually being applied to patients. Yes.
And what should our survivor community know as far as I know you touched on this already, the PARP resistance and BRCA deficient ovarian cancers.
Yes.
So can you just kind of touch a little bit more on anything that they should know about the specific approach in overcoming that?
Yes. So the first thing is, I think we should know is that a lot of people are working very hard as a team on this problem. So this is definitely a very important problem. People are actively working on them now. I think so one thing is really promising now is we have a lot of choices for this second drug to use in combination with PARP inhibitor, and many of them could have the ability to overcome resistance. This is something being actively researched on right now, and from the basic science point of view, a lot of things are looking very promising, but still it will take a little bit more time to really introduce these drugs to the clinic. So people are working very hard on this now, and I think it's a very promising direction. Another thing I think people are actively working on now is immunotherapy. Yes. So PARP inhibitor has shown efficacy in ovarian cancer, and now people are trying with other drugs. Can we target ovarian cancer with other type of cancer therapy? And people are trying combinations as well. So all these new studies are promising. Yeah, maybe not every single one will work out, but some of them will.
Yeah.
As a whole, things are moving forward very rapidly. Yes.
Well, promise and patience, the two of those together, and with a little bit of hope, hopefully we'll keep moving forward there. So with that, I would ask you, what is one message you would give to our survivor community as they're going through treatment for ovarian cancer?
Yes. I think hope. Yes, yeah. Hope is very important. Yeah, I think now a lot of people, a lot of scientists, clinicians, patients are working together. So we know what the major problems are. We're working very hard to address them. And fortunately, so now there are more and more drugs we can work on. People are still identifying novel mechanisms, novel drug targets. So hopefully in the next few years, we will have even more attractive strategies to test. And eventually something will bring a breakthrough to the treatment. Yeah, we will address this problem of drug resistance.
Yeah, well, we're definitely looking forward to a breakthrough. And one last thing, for as far as one other message we want to give our community is ovarian cancer is a silent disease. So what would you say, or what your opinion is on what people can do to utilize an organization such as Ovarcome to raise awareness for ovarian cancer?
Yes. I mean, early detection is very important. And now, because we have a lot more information on the genetics of ovarian cancer, so molecular profiling will become important, because if you know the mutation, for example, in a specific patient, that will really provide the useful information to guide therapy. Because now we have actually drugs such as PARP inhibitor that can direct to a specific patient with specific mutations. So that's something new and something really hopeful. And eventually, when we try to overcome drug resistance, that's the key information we need to get. Yes.
Well, thank you so much, Doctor. We appreciate your time and letting us know, how actively hard you guys are working back there.
Thank you. Thank you for the opportunity to talk to you. Thank you.